That was a great talk, and thank you. And we should move on now to Ksenia Kostanenko, who's assistant professor in neurology, and she's been doing work on brain circuitry in Alzheimer's using optogenetics and multiphoton microscopy. As you may know, Ksenia also runs a nice symposium seminar series now, and it's has some great talks. And so, Ksenia, the floor is yours.
Great. Thank you very much, Jordan. And thank you, Rudy, for inviting me to speak here today. I'm going to start sharing my screen. Okay, can you see the slides?
Yeah.
Okay, fantastic. So the title of my talk today is Improving Brain Circuit Function in Alzheimer's Disease. And as Rudy mentioned, I am an assistant professor here at Massachusetts General Hospital and Harvard Medical School. So in my laboratory, we perform a lot of multiphoton microscopy through cranial windows to monitor what happens in mouse brain. Here is an image that we acquired. Here we can see the blood vessels in red. The neurons are labeled in green, some processes, as well as cell bodies. And this particular mouse is a mouse model of Alzheimer's disease. It already started depositing amyloid plaques, shown here in blue. So here at Mass General, we think a lot about the delicate balance between health and disease. What are the elements that tip the scale towards disease, and how can we reverse these? So the disease of interest in my laboratory is Alzheimer's disease, which is a major progressive neurodegenerative disorder. It is characterized by deposition of extracellular amyloid plaques composed of amyloid beta, intracellular occlusions such as neurofibrillary tangles composed of hyperphosphorylated tau. There is significant cell death that occurs in Alzheimer's disease and can be especially appreciated if we compare the brain of an Alzheimer's patient to that of a control individual. Other molecular risk factors that Rudy and others have identified many years ago, such as APP and presenilins, also the seminal work of Teresa Gomez-Isla and David Holtzman suggested that there are additional pathological features that occur in Alzheimer's disease. Also, the laboratory of my postdoctoral advisor, Brian Baskey, and others suggested that there is more subtle changes that occur in the brain during Alzheimer's disease. And those include disruption of neuronal calcium homeostasis. So what happens is that there is a small yet vulnerable neuronal population that is not able to maintain its calcium homeostasis, and that leads to elevations in intraneuronal calcium. And I will discuss that further in my talk. In addition, there is significant neuroinflammation, that is activation of microglia and astrocytes, these non-neuronal cells that also contribute to the disease progression. So today I'm going to share with you the results of two studies that we conducted a number of years ago now and are published. The aim of the first study is to determine if optogenetic targeting of sleep-dependent brain rhythms alters AD neuropathophysiology in APP-PS1 mice. And the way we monitor sleep-dependent brain rhythms is in vivo in living, breathing mice with voltage sensors. So here is a time lapse of neuronal activity in the cortex of one of the mice. And you can see there's very little activity in this part of the cortex. Over time, it increases and then subsides. And this actually occurs in a very rhythmic manner. So the sleep brain rhythm that I will be talking about today are slow oscillations, and they're important for memory consolidation during sleep. So that's study one. Study two has gained interest recently now that aducanumab was approved by FDA as Aduhelm as the first disease-modifying therapy for Alzheimer's disease. So we partnered with Biogen to determine if treatment with aducanumab, this anti-amyloid beta antibody, leads to plaque clearance and restoration of neuronal calcium homeostasis in yet another mouse model, TG2576 mice. And we use high-resolution multiphoton microscopy to visualize amyloid plaques, shown in white, through cranial windows in mice. So what happens as a result of antibody treatment is what we determined by comparing the same fields of view before antibody treatment and compared those to what happens after. So on to the first study. As the audience, I'm sure already knows, and there have been a number of talks at the seminar series, that sleep is very important, and sleep is disrupted in Alzheimer's disease. Here is an activity pattern of an Alzheimer's patient, and this particular patient was very active in the hours of the night. So what happens is Alzheimer's patients actually very early in the disease progression, during early Alzheimer's disease, or even during MCI, mild cognitive impairment, exhibit sleep disruptions. They have difficulties falling asleep and staying asleep. And what happens is they make up for that lack of nighttime sleep by taking naps during the day. And that further disrupts their circadian rhythms. So slow oscillations were defined by Mircea Stariad in 3 seminal Journal of Neuroscience back-to-back papers as these alterations between up and down states in the frequencies in the delta range, actually, at the frequencies around 1 hertz in humans. Igor Timofeyev later used electrophysiology to monitor these slow oscillations in the cat, and actually did quite a bit of work to determine what cell type is responsible for making these transitions between down and up states, and so on. So what we do in my lab is we monitor slow oscillations using voltage-sensitive dyes. So we're an imaging laboratory, and what we do is we remove the skull from the mouse, as well as its dura, and we load the brains, the cortices, with these voltage-sensitive dyes. Voltage-sensitive dyes bind to plasma membrane and convert a change in voltage into change in fluorescence. So we're basically able to monitor these waves of activity as propagating waves up and down the mouse cortices, as well as between hemispheres. So if slow oscillation is, for example, elicited, let's see, here in the anterior cortex, it would propagate down the cortex as well as to the contralateral hemisphere. So that's exactly what we did. We monitored these slow oscillations using voltage-sensitive dyes, and so that healthy mice have robust transitions between up and down, up and down, up and down states, as can be seen here. And then we compared slow oscillations in our mouse model of amyloidosis, APP-PS1 mice. These mice harbor the Swedish mutation in the APP gene and delta 9 mutation in the presenilin gene. And we saw that slow oscillations are disrupted. They look very, very different in our mouse model of Alzheimer's disease. So then we performed Fourier transform analysis to determine what is it about slow oscillations that is disrupted. And we did so starting at 2 months of age, so very early in this mouse model. So these mice start depositing amyloid plaques around 6 months of age. And at 2 months of age, very early in the disease progression, these slow oscillations were comparable in the APP mice compared to that to the wild type. Yet starting at 3 months of age, we saw a significant decrease in the power of slow oscillations. So power is proportional to the amplitude of the slow waves. So what happens is at 3 months of age, there is significant oligomeric amyloid beta, soluble A beta present in the brains of these mice, and that soluble A beta drops the power of slow oscillations. And that power remains low even during the ages when amyloid plaques are starting to be deposited. Interestingly, the frequency of slow oscillations, in our case, 0.6 hertz, was maintained in APP mice. So what happened was these APP mice still cycle at slow oscillation frequency, albeit at lower amplitude. So then we wanted to verify that it is amyloid beta that is dropping the power of slow oscillations. And to that end, we took a healthy wild-type mouse. We monitored its slow oscillations, and we saw these beautiful, robust slow waves. And then we exposed the brains of these mice to oligomeric A beta preparation. We call it transgenic conditioned media. It was directly introduced to the cortices of these mice right after these traces were recorded. And as you can see, the slow oscillation power decreased as a result of application of these A beta oligomers. So the power was lower while the frequency of slow oscillations was maintained. So this basically mirrored what we saw in our APP transgenic mice, suggesting that it is the amyloid beta that is responsible for the disruption of slow oscillations. So then we wanted to ask the question of what is it about the circuit that leads to disruption of slow oscillations? And the first hint we got was when we did a quick immunohistochemical analysis of cortices of wild-type animals and compared those to those of APP mice. And we stained for GABA, which is inhibitory neurotransmitter. And what we found is that there is decrease in A beta during the ages when slow oscillations are disrupted. We'll later confirm these findings with more sensitive HPLC studies. So then we wanted to see that the the deficits in inhibitory tone were indeed responsible for slow wave disruptions. What we did here was we took an APP mouse that had low power of slow oscillations and applied GABA directly to the brains of these mice. And what we found was that the slow oscillation power was restored. So this suggested to us that slow oscillations are disrupted due to hyperactivity. The circuit is hyperactive within APS1 mice, and the GABA application restores these deficits. So next, once we saw that slow oscillations are indeed disrupted, we wanted to ask this question. Do the slow wave activity disruptions contribute to Alzheimer's progression, or is it simply a symptom of the disease? Is it just an epiphenomenon of what happens to the circuitry, or do disrupted slow oscillations actively contribute to the disease progression? And to answer that question, we used optogenetics, which allows targeting of specific neuronal populations and would allow us to manipulate slow wave activity. So aim one was to determine if optogenetic treatment would restore disrupted slow oscillations and slow Alzheimer's disease neurophysiology in our APP mice. And again, we used optogenetics. The way optogenetics work is we would express a channel rhodopsin 2, which is transmembrane channel, which is light activated. When it is expressed in neurons and activated with blue light, it leads to flow of ions down the electrochemical gradient, and the presence of brief flashes of blue light were able to depolarize the membrane potential. And that would result in firing of the neurons that express it. So what we would do is we would express channel rhodopsin in the cortical pyramidal neurons that Mircea Stariad and Igor Timofeyev determined that this neuronal population is responsible for the slow oscillations, making that transition from down to up state. So then with brief flashes of blue light, we would basically elicit slow oscillations at the frequency that we want. So channelrhodopsin allowed us driving electrical activity of neuronal networks relatively non-invasively with light within intact murine brains. So we express channelrhodopsin in the anterior left cortex, or we express the Cherry marker alone as a control. And then with brief flashes of blue light, we saw that we're able to restore the power of slow oscillations. As soon as the light was turned on, slow oscillations went back to their disrupted state. Thus, we're able to increase the power of slow oscillations while maintaining its frequency. And then we performed this chronic experiment where we used 3-month-old APP mice. We would express channelrhodopsin in the anterior left hemisphere, and then we would express yellow chameleon 3.6, which is a ratiometric calcium sensor. Once expressed in the neurons, it would allow us to determine their calcium concentrations. We would express yellow chameleon in the right hemisphere, and then we allowed the viruses to express. We would perform craniotomies, voltage-sensitive dye imaging to verify that slow oscillations are disrupted in these animals. And then we would install a cannula that would provide guide for the blue light for our channelrhodopsin over the channelrhodopsin expressing neurons. And then at 4 months of age, we would start a chronic light treatment to restore the power of slow oscillations. We would activate the neurons at 0.6 hertz at the frequency of slow oscillations. And we would do so around the clock, 24 hours a day, for 30 days. Before the end of treatment, we would inject methoxyXO4, which would cross the blood-brain barrier and bind to amyloid plaques so that we're able to visualize those with multiphoton microscopy at 5 months of age, and then again at 6 months of age. So we would image amyloid plaques as well as our ratiometric calcium sensor, yellow chameleon. And the working hypothesis was that chronic treatment with light activation of channelrhodopsin would restore slow oscillations in APP mice and lead to slower plaque deposition, as well as normalization of calcium homeostasis. So here is a little introduction to yellow chameleon and how it works. So you probably heard of GCAM, and yellow chameleon is very similar to GCAM. It is also a genetically encoded calcium sensor. The only difference, it has 2 fluorophores. It has a CFP and YFP connected by a linker. Thus, the ratio of YFP to CFP is able to tell us the absolute calcium concentration within neurons within the field of view that we would image. So here, for example, neurons that are shown in blue maintain normal levels of calcium about 100 nanomolar, and neurons shown in red, especially those around amyloid plaques, they have calcium elevations. They're shown in red, and those calcium levels are significantly above 100 nanomolar. So then we would use calcium overload or these calcium elevations as an indicator of treatment efficacy. Once we restore slow oscillations, what happens to the neuronal calcium? That's what we were monitoring. And it is amyloid beta that elevates intracellular calcium. A number of studies were showed that in the APPS1 mice, there are these neurons with calcium overload. And the way we show these neurons with calcium overload are the calcium elevations above a certain threshold. So here, for example, in this mouse model, there were about 20% of neurons with calcium overload. And then also determined that it is the oligomeric beta that is present in TGCM that is responsible for those calcium elevations. So what did we find? We saw that in absence of light treatment, these APP mice continue depositing amyloid plaques. There's new amyloid plaques present in addition to those that were previously there. And if we treat our animals with light activation of rhodopsin to restore slow oscillations, we saw amyloid plaques present. However, there was no deposition of plaques. Does restoring slow oscillations halt amyloid plaque deposition in our animals? And then we looked at calcium overload. And normally these APP mice exhibit calcium overload or these calcium elevations shown in red in this process, as well as the cell body. And restoring slow oscillations prevented neuronal calcium elevations in our APP mice. As you can see, most of the neurons shown here are blue, that is, they maintain normal calcium levels. Thus, by restoring slow oscillations, we're able to ameliorate the disease progression and restore the balance towards health. So next, we wanted to see if increasing the frequency of slow oscillations would exacerbate the disease progression. And here, we basically increased hyperactivity by doubling the frequency of slow oscillations using epigenetics. And we saw that the circuit could indeed keep up with that increased frequency. And we saw that amyloid beta production was increased during the stimulation. And then we performed a very similar chronic experiment where we drove the frequency of slow oscillations at twice the rate between 4 to 5 months of age, again, in APPS1 mice. And what we found was that there was an increase in amyloid plaque deposition when we increased the frequency of slow oscillations. And we also saw an increase in the number of neurons exhibiting calcium overload. So the conclusion so far that slow oscillation power, but not the frequency, is disrupted in APP mice, and optogenetics can be used to manipulate the power as well as the frequency, restoring slow oscillations, halted amyloid deposition, and restored calcium homeostasis, while exacerbating hyperactivity, exacerbated amyloid deposition, as well as calcium homeostasis. So that
Senya, we have 5 minutes left in the session. So if you want some questions, because we have the Mass General research.
Oh, yes.
You might,
sure.
Just so if you want to have time for questions, we have 5 minutes total left.
Sounds great. Yes, I'm finishing up. So disruption of slow oscillation causally contributes to the disease progression, and targeting slow oscillation activity in AD patients might prevent the neurodegenerative phenotypes and slow the disease progression. So now really quickly, I just want to touch on our aducanumab study where we used aducanumab, which is a fully human anti-beta monoclonal antibody in our mouse model, TG2576 mice, with very similar readouts. Let me skip that. So what we found was that aducanumab cleared amyloid plaques in our mouse model. And interestingly, it also restored neuronal calcium homeostasis, as can be shown here. And very exciting for the field, FDA recently granted approval for aducanumab as Aduhelm. Okay, with that, I would like to acknowledge everybody in my group, my collaborators, the folks at Biogen. Thank you for the invitation and our funding sources. With that, I will take any questions.
Thank you, Senya, for really fascinating talk and data. The first question that comes to mind is, I think you seem to be able to explain a lot about what you're seeing because of A-beta's effects in disturbing calcium homeostasis. There's also, as you know, data that says A-beta oligomers block LTP and induce long-term depression. And that's kind of counterintuitive, right? That almost sounds like it should work like GABA. So how do you reconcile those two aspects of A-beta, the LTP blocking and then the calcium dyshomeostasis in terms of your results?
I think it has to do with the type of A beta oligomer, right? They're all different kinds of A beta. For example, A beta monomers are great. They probably have a good physiological function and they're not doing much damage in the brain. The higher is the molecular weight of A beta oligomers, the more toxic we think they are. So I think it has to do with the form that A beta oligomers take.
So you're saying the oligomers may, they drive the NMDA receptor internalization, like Greengard and others showed, and that's inducing long-term depression. But if you have too many oligomers, that may be causing like a synaptotoxicity that causes excess calcium influx. Is that
right? So we see that if we apply this transgenic condition media oligomers, so a beta 42, the oligomers that have a beta 42, for instance, they are more likely to elevate intracellular calcium. So I think it's really the preparation of A beta oligomers, and their identity plays a role.
Yeah, fascinating. Other questions?
Ksenia, I have a lot of questions, but I think we don't have time. I'll catch you up later. Very fascinating talk. Thank you.
Sounds great. Thanks.
And I should say also saying that your data agree very well with Young Kim's 3D culture data, because the main thing we see there is disrupted calcium homeostasis, calcium influx with the excessive amount of A beta oligomer in the 3D cultures. And it'd be nice to think about how we could combine, these different strategies. I know Felipe in our group is doing some optogenetics now. It'd be great to have a little kind of get together group in 114 to talk about that. Yeah. Felipe, any comments?
That sounds great.
Yeah.
No, yeah, it's great. I think it's a great idea, and we should definitely discuss ways to move forward together.
That sounds great.
Cool.
I also just want to make a quick comment. Senya, thank you so much for such a great talk. And offline, I'd be so interested in learning what your thoughts are on the connection between disruption of slow wave activity prior to the onset of Alzheimer's pathology. I mean, there's clearly such compelling evidence from your work that you presented today that there is a connection between the progression of the disease. But I'd love to talk to you about what happens before the onset of plaques.
That sounds great. Looking forward to it.
And I have one key comment. You're finding about GABA receptors. So do you think that has some potential for therapeutics in Alzheimer's?
Yes. We're actually pursuing some of those options. We have an SRA with a company, and we're testing some of their therapeutics. Optogenetics is not such a translational technology, so we want to increase the translational impact by testing therapeutics as well.
Yeah, the comment is that there are multiple natural products that is well known to modulate, especially GABA side. So it's quite exciting directions in my view.
Right. Thank you.
We should get together a joint lab meeting or something on this stuff. And
sure. That sounds great.
That's what these talks are for, getting McCants faculty to collaborate and get new ideas. That's great.
Fantastic.
So we do have the Mass General Research faculty meeting, and I know I need to get to it. Any last comments or questions before we end? These were two great talks, Randy. Ksenia, really wonderful hour here, and I want to thank you for a great job.
Thanks, Rudy.
Yeah.
Thank you.
Great.
Thank you, Randy and Senya.
Bye-bye, everybody.
Hi, everyone.
See you all in 2 weeks.
See you.